Okay, so let's tackle the topic of neurophysiology. And we're going to start out looking at membranes and how they're important in neurophysiology, and then talk about neurons specifically. And I do use figures and diagrams from other sources. I make a point to put the citation for where those sources are, some from my animal phys classes, some from other courses that I've taught. So if you have any questions about the images that I use, please let me know. And of course, I use ones from your book as well. So as we begin, we're going to be talking about basically moving things from outside to inside or inside to outside of a cell, and how a cell, specifically a neuron, can use that to express information about the environment and make decisions within the body on how to respond to what is being picked up in the environment. Okay, so in order to understand any of this, we need to understand membrane potential. It's a very important concept, very important part of this. And all cells, every cell, has a membrane potential. This is the difference in charge between the inside of the cell and the outside of the cell. And all cells are generally going to be negatively charged. They're going to be at a resting negative charge. This is due to the negatively charged proteins and other ions that are inside the cell. In the diagram to the right, this is represented, the negatively charged proteins are represented as anions, or the capital A kind of block with a little negative symbol. So most cells or all cells are at negative when they're resting. Most of the cells in your body are about negative 50 millivolts. Now the book does do a very good job of listing other resting membrane potentials that you can check out and see some differences in different parts of your body. Now, neurons in general have a lower resting membrane potential, and we're going to talk about why that is and how that allows for the neurons to function. So neurons are generally at negative 70. Some of them are at negative 90, which is pointed out in your book as well. And as this says, kind of a clue as to why neurons have a lower resting membrane potential than other cells, the membrane potential, when the cell is at rest, is going to be closest to the ion for which that cell is very permeable. So we will talk about what that means right now. First let's talk about general ideas of how do we refer to changes in membrane potential. And this should probably be review. I'm sure you've seen this before, but if not, when a cell becomes more positive on the inside compared to the outside, so this could be positive ions entering the cell or negative ions leaving the cell, this is depolarization. The opposite would be hyperpolarization. So a cell becomes more negative inside than outside. Again, you could have negative ions entering a cell, or more commonly, as we see in neurons, positive ions leaving a cell. And so the typical types of, the typical form that we see in the firing of a neuron is to have the cell depolarize and then return to resting and then hyperpolarize. So we would call this depolarization, repolarization, and then hyperpolarization. And you also we would also call repolarization returning from the hyperpolarized zone up back to resting membrane potential. Okay. Now, how does all of this change occur? How does a cell become depolarized or hyperpolarized? It's related to the ability of chemicals to diffuse across the membrane. Now, in order for that to occur, you really need channels, so protein channels that will allow for this movement. And there are three ways that these chemicals can move. The most typical one that you've probably studied quite a bit is concentration gradients. So if there's more of a chemical on one side of the membrane than the other, and there are pores or channels through which that chemical can move, then it will move down its concentration gradient. And that would be through passive diffusion or facilitated diffusion, if you're talking about a protein channel. Now, if you move chemicals from a lower concentration gradient to a higher, you're going to have to use energy to do that because the chemicals won't naturally move down their concentration gradient. That's when you get into active transport. But for the purposes of this, we're only looking at straight diffusion. So when a chemical moves down its concentration gradient, that is the chemical diffusion. Now, if that chemical happens to be have an electrical signal to it, so if it's an ion, then it will move in addition to the concentration gradient of the actual chemical, so more sodium on one side than the other, you also have this added parameter of the electrical signal of that chemical. And so it will move from the side where it is a negative charge, in this example, to a positive charge. And so it moves down its, you would kind of think of it as down its electrical chemical gradient. And we are going to look at examples of this to kind of help understand it. So you've got chemical diffusion, electrical diffusion, and then physical. And the physical diffusion isn't as critical. It really doesn't play much of a role in neurons. But it's important for us to mention it here because when we get into the circulatory system or any system where you have some kind of pressure that is going to help to move chemicals, that is a physical movement or a physical diffusion of that chemical. All right, so your book has a nice table showing the general composition of fluid inside and outside of a cell. And of course, these are generalizations. You can get more specific depending on the type of cell. But in general, we are going to have more sodium outside the cell than inside, and more potassium inside the cell than outside. And this is important because it sets up this gradient, both chemical and electrical, to allow for a neuron to fire an action potential or to send a signal. And so I've highlighted the sodium and the potassium here, but these other parameters are also important. Notice the proteins. You have more proteins inside the cell than outside, and they can very frequently carry a negative charge, and that was part of what will provide that negative charge inside of the cell. So in order for any of these things really to move through the cell membrane, you of course need a protein channel. And the picture on the right, the bottom picture, shows a potassium channel. Notice that there is what they term here a selectivity filter. Basically, the structure of this protein are four alpha units, and then these projections out into the center where the hole is to allow for the ion to move through, and they are specific to potassium. So only potassium can move through this channel here. And so channels are very specific to their ions or to, for example, water. You have aquaporins that are specific to water, and other types of chemicals, glucose, anything. So now the only exception in this table to the left would be your oxygen and your carbon dioxide. That is going to simply diffuse across the cell membrane. It doesn't need any special channels to do that. And then the upper picture, this is from your book as well, and it goes over, it shows how quickly diffusion can occur, and your book discusses that. And to give you an idea that this speed at which a chemical is moving within a fluid allows for the neurons to respond very quickly, or any cell to respond very quickly, and therefore to function properly. Okay, so the simplest case when we're looking at potassium is in terms of the electrical signal. Potassium is going to diffuse down its concentration gradient. So if we're only looking at potassium, and of course this isn't going to happen in nature, you're not going to have a cell that is solely allowing potassium or any other ion to be contributing to its chemical and electrical properties. But this should give you an idea to help you understand what we're talking about when we're talking about this electrical changes that occur, and specifically Nernst potential. So when potassium diffuses down its concentration gradient, it does this until it reaches its electrical potential. So you wind up with, again, more potassium inside of the cell than outside, and that results in what we call an equilibrium potential, or we call that capital EK. So the equilibrium potential of potassium, or the equilibrium potential of any other ion, is going to contribute to the overall membrane potential of a cell. And so if we can calculate, knowing how much potassium is inside the cell versus outside the cell, we can calculate the Nernst potential, or the electrical contribution to these gradients that is provided by potassium. And so when we do this, we get about negative 94 millivolts. So if a cell is only permeable to potassium, then the membrane potential would be negative 94 millivolts, or the membrane potential. So let's look at sodium. So sodium is the opposite. It is it has a much larger draw to move outside the cell than inside. And so the equilibrium potential of sodium is the opposite of the equilibrium potential of potassium. And again, we're looking at this in terms of if we're only thinking about sodium and no other ions or no other chemicals. And so if we look at the Nernst potential of sodium, therefore if the membrane was only permeable to sodium, then the membrane potential would be positive 61 millivolts because of this increase in sodium outside of the cell versus inside of the cell. And so I think you can already see we've got a negative 90 or so for potassium, a positive 60 for sodium, and in order to truly understand how the cell is functioning and the membrane potential of the overall cell, we have to look at all of the ions and all of the chemicals that are involved. And so we do this using the Goldman equation, also called many other things, but I've always just referred to it as the Goldman equation, which I realize as a scientist is kind of leaving out the other people that were important to figuring that out. But so this is an equation by which you can calculate the membrane potential off of the permeability and the concentration of the ions that are involved, or of the chemicals, any chemicals that are involved. Now, of course, we simplify this because there are some chemicals for which there's no impact to the membrane potential, and they're neutral. There are some chemicals for which there are no channels, and so the permeability of the cell is very low, and so it wouldn't contribute anything to the calculation of the membrane potential. And as you can see here, we can compare, we are comparing notice where the outside and inside ions are. We are comparing the overall contribution of positive charge outside the cell on the top of the top equation there, and overall positive charge inside the cell on the bottom. And then the opposite is true of the bottom equation. That would be calculating the overall negative charge outside and inside the cell. And I put here the membrane potential of neurons, again, is somewhere between negative 90 to negative 70, and that depends on the type of neuron that you're looking at. Again, this would be how you calculate, how you get to the resting membrane potential of a cell. And notice it's all related to permeability and the concentration of the ions that you're interested in. Okay. So when we look at this on a scale, we notice that the resting membrane potential of those neurons is very close to the membrane potential that would be the equilibrium potential that would be supplied by potassium. So if membrane potential is based off of the concentration and the permeability of ions, then by looking at this, I hope that you can see, well, neurons must be much more permeable to potassium than to sodium because the membrane potential is so close. Basically, it's easier for potassium to move down its electrical concentration gradient and reach its Nernst potential, or get closer to reaching its Nernst potential than sodium, and so we get a resting potential of -90 to -70. And this is important because this resting potential, this resting membrane potential, is what the neurons use to change, to send a signal. So when a neuron fires an action potential, it is basically moving the electrical signal of the cell more positive, so closer to the sodium Nernst potential. And so this resting setup of the cell allows for the cell to be primed to send a signal when it is stimulated. So let's really briefly talk about why is the cell so permeable to potassium. Now, we know why because of big picture, like this is why the cell, but physically, why is the cell so permeable to potassium? It's because of the channels. So potassium leak channels are probably the most obvious way that potassium is able to move from the cell. This allows the potassium to literally leak out, and these are open all the time, specific to potassium, and they do not they don't close, they don't need anything to open, they're simply always open. Now, the leaking of the potassium is important to the cell. It helps return it to rest, and it also it helps with the something that we'll talk about later, the relative refractory period, but it also contributes to the permeability to potassium. And then the sodium-potassium pump also contributes to the permeability of potassium, and of course sodium as well, but we're only talking about potassium here. So these two channels together contribute quite a bit to the permeability of these cells to potassium, and therefore allow for the increased pressure on the, the increased equilibrium for the cell towards the Nernst equation, or equilibrium equation for potassium. Okay. So let's also talk about gated potassium and sodium channels. Now, the ones that we're talking about for the neuron are gated by voltage. So these are the voltage gated. But gated channels, or channels for sodium and potassium in general, the way that they are specific to these ions would be through, again, you've got those selectivity filters in the potassium channels, but the other thing with sodium channels especially is that sodium is smaller than potassium, and that's shown in the numbers in the bottom left here. It's smaller than potassium when it is dehydrated. And what do I mean by that? If you look at the diagram in the upper left, that blue circle is the sodium ion, and the red and the white circles around it are water. And so ions are going to attract water. Both potassium and sodium are going to attract water. And the way that the sodium channels help to allow only sodium to pass through is that as the sodium approaches, it is the properties of the channels allows for the removal of the water, or the dehydration of that shell, that hydration shell around the sodium. And then the size of the pore only allows sodium through and not potassium. And so these gated sodium and potassium channels are extremely important, both in terms of selectivity to what passes through, but also when. So if a The channels we just looked at, the pump, sodium potassium pump, and the leak channels, those are constantly functioning or constantly open, versus these gated channels only open under certain circumstances, whether that be voltage gated, which is shown in the upper part, or some other type of gate that will allow for the opening of the channel, whether that be binding of a ligand, or if you're talking about like touch receptors, it might be a mechanical gate. Okay, so here again, the ones that we're really interested in when we're talking about gated channels in the neuron are voltage gated and ligand gated. Again, voltage gated channels are going to open when the voltage reaches a certain point. So the voltage is high enough inside the cell to cause the channels to open. And think about how can you increase the voltage or the electrical property inside of a cell? How can you make a cell more positive? You could hit it with an electrode, hit it with an electrical signal, as we do in the laboratory. And so the image on the bottom left here is showing hitting a channel with a stimulus and the opening of those sodium channels from when that stimulus, from the opening of the stimulus. And also the voltage, that diagram also shows how the voltage gated channels can be opened and closed very quickly if the stimulus is small enough, or if it's a really big stimulus, if there's a lot of positive charge in, they could be open for a longer period of time. So those are voltage gated channels. But in a natural setting, we don't have electrical currents, meaning electrodes that we can insert into our cells like we do in the lab. And so the cells will change the voltage by changing the ion concentrations. And this is how a neuron fires an action potential. It opens its other channels, its ligand-gated channels, to allow sodium or other positive ions, but usually sodium, to enter the cell. That increases the voltage of the cell. So you have a ligand-gated channel, like a sodium channel, open. Sodium goes into the cell. It makes the cell more positive, and that causes the gated channels to open. So the voltage-gated channels and the ligand-gated channels are intertwined in the initiation of a signal that's sent by a neuron. And then it is the opening of the voltage-gated channels as you move down the neuron, down the axon, that allows for the other voltage-gated channels to open. And I will show you a diagram and talk about that more in a minute. The other thing that this slide shows is the way that we measure this in a lab. So this is a diagram, the bottom diagram in the middle is showing patch clamping. Basically, you can attach a very small pipette to a cell and remove or measure the movement in a single channel or a series of channels, depending on how large the pipette is and how good you are at targeting those channels. So patch clamping means you patch onto a small area of a cell a pipette or a clamp, and more frequently, to have better control over the ions and the flow of electrical signal, you remove, so you would tear off that channel, kind of use suction and vacuum to tear off the channel, and then put it into a beaker with a solution for which you know the levels of ions in it. And so that's what's shown in the right picture there, in the bottom right picture, and then the top of that picture is showing recording in a cell. And notice that you're always going to have a ground that is measuring the natural basic overview of the electrical signal, and then you can measure the changes that are caused by or result from the actual channel itself, and so that would be the other electrode there. And your book talks about this, so if my explanation is confusing, definitely read it in your book. Okay. So when we're talking about action potentials, these are the use of the neuron of the gated channels and the gradients that were set up, both chemical and electrical, so the electrochemical gradients that were set up by the sodium-potassium pump and potassium channels to propagate an electrical signal down an axon. And an action potential is basically very plain, straightforward, depolarizing the cell past a threshold that brings the cell closer to the sodium Nernst potential. So notice that in the overshoot there. And again, allows for the passage of that information down the axon. And we'll talk about how that information is passed on from an axon to from a neuron to other cells in a moment. So the diagram on the right is showing how we would measure this, and basically the movement of the membrane potential as you move down the cell. So you're going from a negative to a positive down the cell. Now, I think that this diagram will help a lot because I've already mentioned the axon and movement, propagation of an action potential down an axon. And so I wanted to include this to give you an overview of basically the anatomy and of the cell, the neuron, and how that relates to the function of that neuron, or the physiology, which is what we're really interested in. So the ligand-gated channels that I mentioned that will result in the voltage-gated channels opening, those are going to be located in the dendrites and the cell body, or the soma, of the neuron. And remember, when we're talking about neurons, we're talking about potentially really big cells. You have a dendrites and a soma, or cell body, of a neuron in your spinal cord with an axon that reaches all the way down to your pinky toe. And then your axon terminals would be at the muscles, the muscles of your pinky toe. So these cells can be very large, but they do have very distinct functional units or functional zones within that one cell. So the dendrites and the cell body are where the signal comes into the cell, and this makes up what we refer to as an input zone, or a zone that receives the information. So the olfactory neurons that you have in your nose that pick up on odorants in the air, those are extensions of a dendrite, and they have channels that are very specific to certain odorants, and when you smell something, what happens is the odorant binds to that ligand-gated channel, and that causes an action potential to be sent down the axon and into your brain. So you have the dendrites in the cell body which pick up on the signal. If the signal is strong enough to trigger an action potential, this would occur at the axon hillock. That's the area of the neuron that leaves from the cell body and extends out and down the axon. So kind of the gate to the whole action potential being fired down the neuron is at this axon hillock, or the trigger zone. If you get enough of a signal, enough of an increase in membrane potential that it triggers the opening of those voltage-gated channels in the axon hillock, then you're going to have an action potential, no matter what. Action potentials are all or none, so they're going to fire whether or not, once they reach threshold, they're going to fire. The action potential itself is propagated down the conducting zone, or the axon, that, as this shows, can be very small, a millimeter, or very, very long, up to a meter long, depending on how tall somebody is, or how long their legs are. The axon ends at an axon terminal, which is the neuron, this cell's side of what we call the synapse, or where the signal is passed on to either the muscle or the other neuron or a gland to continue that signal on. So the end goal here, if there's enough signal in the dendrites and the soma to trigger an action potential, you're going to get release of neurotransmitter at the axon terminals. Of course, there are different types of neurons that will respond to different stimuli. So we categorize these in general as afferent neurons that pick up on sensory stimuli. That could be outside the body, like the odorant example I gave earlier, or inside the body, monitoring CO2 and pH levels, for example. And so you have these sensory neurons that are bringing information about the environment, internal or external, to the brain and spinal cord, or the central nervous system. And we call the neurons in there the interneurons. Now, we will look in a moment at the complexity of synapses between neurons and how you can have lots of cells synapsing on one cell. So, of course, this is a very, very simplified version of what's occurring. But in general, you have afferent sensory neurons bringing information into the integration center, or the brain and spinal cord. And then if there is a response needed or triggered, that will occur through the efferent neurons, or the motor output in the peripheral nervous system. So when we talk about the dendrites and the soma and the channels that are picking up on stimuli, that can vary depending on which type of neuron you're talking about. Most of these are going to be ligand gated, because most of the neurons are interneurons like the Purkinje cell, or motor neurons that are going to respond to neurotransmitter. So those would all be ligand-gated channels, and in your sensory neurons, if they are also responding to chemicals, like your taste buds or your olfactory neurons, they are going to also respond to ligands. Now, there are exceptions, of course. In the sensory neurons, you've got your cells of your eye that respond to light. You've got your ear and your touch receptors that respond to changes, mechanical movement. But in all of these sensory receptors or sensory neurons, there is a channel that will be triggered to open that will depolarize the cell and send an action potential if there is enough signal. So again, you have afferent synapsing on interneurons, and then interneurons can synapse on each other, and eventually they will synapse on an efferent neuron, which will bring the signal out to a muscle or maybe to a gland if you need some hormones released. So let's start. We're going to start in the signal coming in the dendrites in the cell body, and then work our way through the neuron. So when you look at dendrites in cell body, you're looking not at action potentials, but at what we call graded potentials, because they have a grading to them. The concentration of the chemical that we're interested in this case sodium, is going to be greatest around the channel itself, and then become less and less through the process of diffusion in the cell. So you have an incoming signal. This is, let's stick with ligands. So you have a ligand, a neurotransmitter, that binds to a channel, a ligand-gated channel, that causes the channel to open, and you have sodium entering the cell. This would occur again in the dendrites or the cell body and create a graded potential. Now, the change in membrane potential is this graded potential. It is the change in the more positive signal in this example inside the cell versus outside, again, changes the membrane potential of the cell. So this is what's occurring throughout the dendrites and the cell body of the neuron. And the picture that I have in the upper left-hand corner is a summary of this in terms of losing signal through both resistance, and then the bottom picture is showing how you get this decrement of the signal as you get away from either the channel, or if you're actually measuring something, it's going to be the electrode that you're getting away from. Now, in order for an action potential to be expressed, you need a opening of the voltage-gated ion channels in the trigger zone of the axon hillock. So imagine the graded potential that's occurring in the dendrites and the soma, the cell body. And then the axon hillock coming off of that. So that's what's shown in the picture on the bottom right. You have excitatory postsynaptic potentials. That's what EPSP stands for. So those excitatory depolarizations that are occurring in the cell body and the dendrites, but they're not high enough or close enough to the trigger zone in the axon hillock to cause an action potential. And so, if you have more than one channel opening, or if you have a channel opening very, very close to the trigger zone, that is going to be more likely to hit the threshold and cause an action potential. And what do I mean by threshold? This is the change in membrane potential, in this case -50, that causes those voltage-gated sodium channels at the trigger zone to open, and that is what propagates down the axon as an action potential. So when you hit that threshold, you get, as this says, an explosive depolarization that is all or none. It's going to go down the axon. The none would be these EPSPs that are too far away or too weak to trigger the action potential. After the action potential, of course, you get repolarization and it returns to rest. And the picture to the bottom left is from Wikipedia, actually, and it's simply showing the movement of the change in membrane potential or action potential as it moves down the axon to reach the axon terminal. So now we have the signal coming in at the dendrites and the cell body. If it's a strong enough signal, or it's if you have enough channels opening to change the membrane potential enough to hit threshold, that will occur at the trigger zone on the axon hillock, and then you get an action potential that propagates down the axon. So I've already kind of alluded to this, but I wanted to show you a detailed diagram of it. There's two ways that you can reach this action potential threshold. You can spatially summate, so you can get more than one channel opening throughout a cell to cause the threshold to be reached. So that would be spatial summation. And more typically, you're going to have many channels that are opening, not just two. But this is a good example of, if you only have one channel opening, you're only going to get up to -60. If you add that on, if you add that together, you hit the threshold of -50 and you get an action potential. So that would be spatial summation. We also have temporal summation. So this would be one channel getting hit with a stimulus close enough in time that the membrane hasn't returned to rest, and so it adds on to itself. Typically, you're going to have both temporal and spatial summation. So you'll have multiple channels potentially opening multiple times in a row to allow for the cell to depolarize and hit threshold. But again, you can have either or both spatial summation and temporal summation. So again, temporal would be the same channel opening twice in close time period, close distinction to reach threshold. And why does threshold have to be reached? It has to be reached to open the voltage-gated channels. It will open both the voltage-gated sodium channels, which cause the cell to become more positive. It changes the permeability of the cell to allow the cell to approach the Nernst potential of sodium, versus the voltage-gated potassium channels would have the opposite effect. However, the voltage-gated sodium channels open before this potassium channel, so faster, and that is what causes that depolarization, and then it is the voltage-gated potassium channels that help to repolarize the cell. The other thing to notice here is in the sodium channels, there's a gate there, a little inactivation that occurs that physically closes the channel with that little extension of the protein. It's labeled here as slow closing triggered at threshold. So that closing of that gate, that physical closing, prevents sodium from entering the cell. Even though the channel may be open, it is blocked by that gate. And that is very important. We're going to talk about why that is in a minute. So as the signal moves down the axon, we call this propagation of the action potential. What it does is the opening of the initial voltage opening of the sodium channels causes sodium to enter the cell becomes more positive, that changes the membrane potential in the neighboring areas of the membrane, which causes those voltage-gated channels to open. So you get this propagation down the axon. Now, notice that in the image to the right, it's showing that moving in both directions. And this is important because this could occur if the sodium channels didn't have that little kind of extension of the protein that inactivates the channels. So we'll talk about that in a moment, because we want the signal to move one way down the axon. Now, how do we get this? I think I've already touched on this, but you can have one large graded potential right near the trigger zone that can cause enough threshold to be reached right at these voltage gated channels to cause the signal to move. Or you can have many graded potentials throughout the cell, so many receptor sites or different types of receptors that open and help to depolarize the cell. Something else to remember is that these potentials don't necessarily have to depolarize the cell. Some of them do hyperpolarize, so some of them do open the potassium channels, cause potassium to leave. And so that hyperpolarization versus depolarization, hopefully it's pretty obvious which one will cause an action potential and which one will make an action potential harder to occur. Okay. So after an action potential, ion channels are inactivated and also deactivated. So these sodium channels, that little protein that plugs the sodium channel, inactivates the channel, and therefore prevents it from opening, even though the voltage might be changing due to the other sodium channels that are open. So it prevents, in the previous slide, it would prevent that signal from going to the left, and the signal would only be able to go to the right. The other thing that occurs after those sodium channels close is the potassium channels get deactivated. Notice that they stay open longer than the sodium channels. They open later, and then they stay open longer than the sodium channels. What happens to a cell when a potassium channel is open? Where does the potassium go? It leaves the cell. When a positive signal like potassium leaves a cell, it hyperpolarizes that cell. So by having a longer period of time for which the potassium channel is open, you are going to hyperpolarize that cell. And again, that is important for the function of the neuron. So when you get these changes in conductance, or the movement of the ions, that will affect the changes in voltage as well. Initially, you're going to have the sodium channels open from the voltage change due to graded potentials. That's going to cause the sodium channels to open. Sodium's going to enter the cell. It's going to bring the cell up towards Nernst potential of sodium. It's going to make the cell much more positive or depolarize it. So that's 1 and 2 there. Then the sodium channel gets inactivated, so that's 3. That's going to mean you have no more positive signal coming in, but now your potassium channel is open, you have positive signal leaving, so that's going to bring the cell back down to rest and hyperpolarize it. Notice where it says kind of positive after potential, meaning you're hyperpolarizing the cell by having that having that potassium leave the cell. And so you get this extensive conductance of potassium over the course of the action potential that again hyperpolarizes the cell. And that's what's shown in the bottom graph on the left. This is a summary from the Animal Phys textbook that I use in another class, and so it again shows the same thing, just in a different way. It might be easier for you to understand. So if you want, look this over. Basically, I already covered all of this, so I'm not going to talk about it again. But notice the depolarization and then the repolarization followed by hyperpolarization. So the reason that the sodium channels get inactivated is, again, to prevent the signal from going backwards in the opposite direction down the axon. So backward current cannot, while it can open the sodium voltage-gated sodium channels, you cannot have sodium enter through them because of that inactivation that occurred. So you're still going to have this spreading of voltage out from the channels, but it can only trigger the opening of voltage-gated channels down the axon. And so the action potential can only propagate in one direction, down the axon. And that is what is shown here. And then, of course, the repolarization of the cell is occurring because of the potassium channels. So if you look to the left of this diagram, where the dotted lines are, and it's changing from, instead of being positive inside the cell during the action potential, it goes back to being at rest negative. That return to negative is occurring because of the potassium channels being open. And remember, throughout all this, we have those sodium-potassium pumps functioning. Now, this closing of the sodium channels, or the inactivation of sodium channels, and the elongated opening of the potassium channels causes what we call refractory periods. The absolute refractory period would be when the sodium channels are closed by that inactivation. And so no amount of signal or positive signal depolarization, no amount of depolarization can cause those sodium channels to open, and so nothing can cause an action potential. And so I like to say that during the absolute refractory period, absolutely nothing can cause an action potential. The relative refractory period is the amount of time where the cell is hyperpolarized. And so the cell is in a refractory period, meaning it is much more difficult for that cell to fire an action potential, but it is possible. Think about the fact that the cell is hyperpolarized at this point during the relative refractory period. And so it can reach threshold, it's just going to be harder for it to reach threshold and therefore to send an action potential. So if the signal is strong enough, say you smell something really strong, and so you have lots of odorants binding to those channels, and so the cell is very, very depolarized, that is going to overcome that relative refractory period and send a signal in close succinction, very, very close to the previous one, or send an action potential. So if the signal is very strong, the neuron will send multiple action potentials in a row, overcoming the relative refractory period and communicating to the interneurons that it is synapsing on that this is a very strong signal because I'm able to send action potentials in close succinction, very close together. So we've talked about action potentials quite a bit. I just want to review very quickly the properties. So I've mentioned they are all or none. Once you hit threshold, they're going to go, meaning all. If you don't hit threshold, they're not going to go, so none. Depolarization is going to cause them. So if a cell is hyperpolarized for any reason, it's going to make it harder for that cell to send an action potential. They always reach the same amplitude. They don't summate, meaning they're not going to add on to each other. The triggering of an action potential can result from summation of graded potentials. So graded potentials can summate, but action potentials cannot. It is the frequency, not the strength or the amplitude of the action potential that is important. And they have a constant conduction velocity, so they're going to have a consistent velocity at which the channels can open and close and reset for any given cell. And that velocity is dependent on the diameter of the cell. So the bigger the diameter, the faster that cell can fire an action potential, and whether or not the cell is myelinated, which we'll talk about in a moment. If a cell is myelinated, as you can see in the picture to the right, you're going to get a much stronger, a much faster action potential. Okay. So we've touched on this. I mentioned the decrement. Voltage is going to decrease with distance from the signal, whether that be an open channel, or if you're a scientist working in a lab, it might be an electrode that you're causing a signal with. So we call that conduction with decrement. That's that, on the graded potential slide, I showed that picture of the decrease in sodium as you get away from the channel. There is a high resistance created when you have the juxtaposition of intracellular and extracellular fluids and a membrane separating them. And you also get resistance from the channels themselves. The membrane can cause a decrease in voltage because of those leak channels, so you're always going to have some charge leaking out. So what this is referring to is having a membrane is going to decrease the voltage along the axon. And so when you have those leak channels and you have that exposed membrane, that's the important part. It's that the membrane there's nothing separating the membrane from the intracellular and extracellular fluids on either side of it. That's the key. That is going to decrease the voltage and, again, slow down the transmission of the signal. So, as I mentioned, the way that neurons can overcome this is by being larger in diameter. That's going to allow for the signal to be conducted quicker. However, the bigger diameter axons are big, they're going to take up space, and they're expensive. Your cells are going to have to create all that cytoplasm and membrane and everything to build the cell. And so vertebrates, specifically humans too, especially humans because that's what we're interested in here, use myelin to allow for this very quick conduction down the axon. So myelin is equated to the insulation of wires. So myelinated fibers are like a wire in the picture to the bottom right, coated in something that prevents the loss of signal along the wire. So by having myelin surrounding the membrane, you separate the membrane from that extracellular fluid. You keep the signal inside the cell instead of allowing it to leak out or allowing it to hit the resistance that's created by that juxtaposition of intracellular and extracellular fluid. Now, myelin is created by two different types of cells depending on where you are. It is in the central nervous system, oligodendrocytes create myelin. And in the central nervous system, as you can imagine, there are much more neurons closer together, and so oligodendrocytes can myelinate multiple cells, versus Schwann cells in the peripheral nervous system, which you tend to see multiple Schwann cells associated with one neuron throughout the length of that axon. The regions in between the myelin where the signal is able to be propagated are known as nodes or nodes of Ranvier. And then the area in between where you actually have myelin would be internodes. So the nodes are about 1 millimeter apart, depends on the neuron, of course, and there's lots of those voltage-gated channels there to kind of reinvigorate the signal the action potential that's propagating. So this is what it would look like in the peripheral nervous system especially. You've got areas where the myelin is coating the outside of the cell membrane of the neuron, of the axon of the neuron, and regions where it's not present, where the signal can be propagated because you have those gated channels in those nodes of Ranvier. And the purple in here is representing the nucleus of the Schwann cell, and then the orange wrapping around the axon, that would be the myelin sheath. And it is a fatty substance. It's like a lipid-rich sheath that surrounds that cell, neuron. So when we're looking at the regions where we have myelin, we don't have gated channels there. We don't have channels there. It's just myelin keeping the signal inside the cell. And then where we have the nodes, that's where the channels are concentrated. And then this is a diagram showing what it would look like in the central nervous system, where you have oligodendrocytes myelinating multiple cells, multiple axons of multiple cells within the central nervous system. And if it's a really short axon, you might have just one oligodendrocyte myelinating it, or if it's a longer one, you'll have multiple ones. But we still have the myelin sheath, and we still have the nodes of Ranvier. Now, the nodes of Ranvier are a little different between the central and peripheral nervous system, but we will probably touch on that when we get into the nervous system in general. We refer to this movement of the action potential down the axon when we're looking at myelin as saltatory conduction. And so this diagram from, again, the Animal Phys textbook, I like it a lot because it shows the channels concentrated in the nodes. And I love diagrams. They always help me. And then you can see in the picture on the right, the top part of that picture, the sodium is going to enter the cell when the action potential reaches that node, and that is going to move down the axon and cause the voltage-gated channels at the next node to open, and then the signal will come in and then move down the axon. And remember, we still have inactivation of sodium channels and the elongated opening of potassium channels. So we're still going to have hyperpolarization of the cell and typical appearance of action potential. And so the books frequently refer to, and even this picture shows the action potential leaping from node to node. That's what saltatory means. But for me, that always confused me because it made me think that the signal was outside the cell, meaning the membrane potential was changing outside the cell. But it isn't. It's inside the cell where you get this change in membrane potential. I would prefer if those blue arrows went through the axon and then showed the signal changing at the nodes. I like this diagram because it shows how action potentials would appear in the way that we are used to looking at them in a non-myelinated versus myelinated cell, or axon, I should say. And I think that it is a very good representation of how you save time and energy, really, because you're not having gates open as much with myelin. And therefore, the energy that the oligodendrocytes or Schwann cells are spending making the myelin and maintaining it is useful in terms of allowing that action potential to move down the axon quicker and more efficiently in myelinated neurons versus non-myelinated. You can imagine that if an axon's very short, then non-myelinated might be better because you don't have to myelinate it. You don't have your body isn't going to use energy for the oligodendrocytes to myelinate it. But the longer the axon, the faster you want the signal to move, the more likely it is that it needs to be myelinated. Now we've reached the end of the axon, or the axon terminal, the fourth functional zone of the neuron, and I want to look at synapses. And we're going to start looking at neuron-to-neuron synapses, and then we'll look briefly at neuron-to-muscle synapses. And there is a third. You do have neurons that synapse on glands as well. It's very similar in terms of what happens, whether it's synapsing neuron-to-neuron, neuron-to-muscle, or neuron-to glands. You're going to have the action potential reach the end of the cell, or the axon terminal, and that is going to trigger a different voltage-gated channel, voltage-gated calcium channels to open, which causes the release of neurotransmitter. This diagram shows, and this is also from the Animal Phys textbook. I realize I didn't put my little note there. This shows how one neuron in the picture, in both of these pictures, shown in green in the picture to the right and in orangey yellow in the picture to the left. One neuron can have many, many, many synapses on it, or many axon terminals synapsing with the dendrites and the soma. So we're kind of back to where we started. We're back to looking at releasing neurotransmitter and causing potentially another action potential at this axon because of the gates and its dendrites and its soma. So in neuron-to-neuron synapses, we're talking about chemical synaptic transmission. That would be as opposed to gap junctions, which would be a direct electrical current that moves back and forth. The vesicles that are in the axon terminal, or the presynaptic neuron, contain neurotransmitter. And it is the opening of these voltage-gated calcium channels. Calcium comes in, it binds to the vesicles and causes them to bind to the membrane at the synapse and exocytize and release the neurotransmitter. This is one example, the most common example. There are other mechanisms by which this occurs, but this is the classic example. So voltage-gated calcium channels open, calcium enters and causes the exocytosis of those synaptic vesicles, which contain the neurotransmitter. You may or may not automatically recognize this, but of course the neuron and up at the soma has created these synaptic vesicles containing the neurotransmitter and then transported them down through microtubules or the cytoskeleton to the synapse. So remember that the neurons aren't just passing the signal along electrically, they're also creating the neurotransmitter and passing that out through the synaptic vesicles. This is the example of, again, from that animal phys text, of the neuron-to-neuron transmission of signal. And so it shows diagrammatically the one there, those blue arrows, that would be the action potential moving down the axon. Once the depolarization reaches those voltage-gated calcium channels, they open, calcium enters, and they cause the vesicles to bind with the membrane to release the neurotransmitter. And remember, neurotransmitter would open those ligand-gated channels, which will allow potentially sodium to enter the cell, or perhaps allow chloride to enter the cell, or potassium to leave the cell, which would cause the cell to become hyperpolarized. So you can have both depolarization or hyperpolarization, and that would all depend on the receptor. What the receptor is specifically going to allow to enter or leave the cell. Again, that gets into that specificity for either sodium, potassium, or chloride, or whatever that channel protein, that receptor, is for. Okay. This is the neuromuscular junction. So the major difference here is we know we're talking about acetylcholine as the neurotransmitter. That's going to bind to these channels predominantly, which are allowing sodium to enter the cell, but they're also allowing potassium to leave and therefore depolarizing the cell. We will talk in a couple weeks about how the contractile elements then are triggered to contract. So sodium's entering the cell, depolarizes the cell. That depolarization gets spread kind of like an action potential, sort of, down the membrane. That's shown as 8 with those little blue arrows to the left and the right, and that triggers other voltage-gated sodium channels to open. So we've got ligand-gated sodium/potassium channels opening due to the acetylcholine, and then we have voltage-gated sodium channels opening, also helping to depolarize the cell. And that's where we will leave it here with neuromuscular junction until we pick it up when we get into the muscular system. Okay. Now, as I said, a neurotransmitter can cause depolarization, which we've looked at quite a bit, or it can cause hyperpolarization. And we refer to these as, if it's hyperpolarizing the cell, that would be inhibitory. So this would be inhibitory postsynaptic potential. If it's exciting the cell, that would be excitatory. If it's depolarizing the cell, it's excitatory. That's excitatory postsynaptic potential. So you should be very familiar with these IPSP and EPSP. The IPSP is going to make it harder for the cell to fire an action potential. The EPSP is going to make it easier. And that's demonstrated here. So an excitatory synapse will depolarize, bring the cell closer to threshold, whereas inhibitory brings it farther away. And you can, this all depends on the type of receptor, whether that receptor is going to allow a negative ion into the cell or a positive ion out, or and whether that receptor, what opens that receptor, the type of neurotransmitter that causes that receptor to open. And this diagram shows very well how you can have both excitatory and inhibitory postsynaptic potentials occurring on one cell. So you can get, this is where that integration comes into play. So in the central nervous system, you're going to have integration from multiple sensory neurons onto these interneurons. And then, so if you get enough excitatory, as is shown with the spatial summation there, if you get multiple excitatory channels opening, or you get one excitatory channel opening multiple times, then you're going to have a summation of that depolarization and you'll get an action potential. But if you also get input of inhibitory, that can cause it to be harder for the postsynaptic cell to fire an action potential, and you might cancel out your excitatory postsynaptic potentials. So you can see here, just with this very simple example of one postsynaptic cell and 3 presynaptic cells, that we've got a complicated system here. And so the nervous system is very complicated. We have both convergence, lots of neurons synapsing onto one neuron, and divergence, lots of branches coming off of one neuron, or lots of axon terminals synapsing on many other cells. So this would be just thinking about neurons within the central nervous system talking to each other. Then you add in the idea of multiple sensory neurons all synapsing on multiple cells within the brain, and you can see that it is very, very complicated. And this is how our brain is able to do such complicated things and integrate so much of the information about our environment into it. And this is a diagram representing that, and I would even say this is a simplified representation, because as you saw in the previous slide, you can have hundreds or thousands of these synapses occurring involving what we would call one postsynaptic cell. So you can see that each of these colors is representing a different neuron. Some of them are positive, some of them are excitatory, some are inhibitory, and then that single cell is synapsing on multiple other cells. And on some of those cells, the release of that neurotransmitter might cause an excitatory postsynaptic potential if it has a certain receptor, and then if other cells have a different type of receptor, it might be inhibitory. So again, very complicated. This is something that your book touches on. I wanted to bring up because we are talking about the real world and the types of recordings that you might see if you were a healthcare provider or when you go to the doctor. So when we measure action potentials extracellularly, we're actually measuring we're not measuring inside the cell, we're measuring next to or near the cell. And so those recorders pick up on the stimulus, meaning the electrode or the electrical signal that is causing the action potential, causing the recording of the action potential. So we would actually see a bipolar action potential, meaning first measuring the stimulus and then measuring the actual action potential itself. And so you can see this represented as what's occurring in the cell in the pictures to the right here, and the movement of that action potential down the axon as represented by what we actually would measure, which is in the picture to the left. The last thing I want to touch on is a review and covering of the different types of cells within the nervous system. So the picture is showing central nervous system, but we also have similar cells in the peripheral nervous system. So in the central nervous system, of course we have neurons. Neurons are their own cell. Everything else that supports the neurons we refer to as glial cells. So these glial cells are astrocytes, microglia, ependymal cells, and oligodendrocytes. We've already talked about oligodendrocytes and Schwann cells. They make myelin. Astrocytes provide the blood-brain barrier, so they create tight junctions and a tight covering across the capillaries of the brain to prevent certain chemicals and certain things that are in the blood from entering into the brain. So astrocytes do the blood-brain barrier. They also provide support, physical and chemical support within the brain to the neurons. And they you can see how the astrocyte in the picture kind of extends its foot processes out and connects the cells to each other. So those astrocytes are really providing the structure, keeping the neurons in the right placement and keeping all of the cells kind of physically connected. Microglia are the immune cells of the brain, so they are a derivation from monocytes in the or white blood cells, and they would be the macrophages of the brain. So they go around and kind of monitor the brain. If there's a pathogen, they would respond to it. So if you see lots of microglia in one area of the brain, that indicates an infection of some sort, or they can also respond to physical changes. So like if you get a bump in the head and a concussion or something like that. And lastly, ependymal cells, those are the green cells in the upper part of the picture. They are what create and monitor and line the cerebral spinal fluid containing parts of the brain. So the ependymal cells, you would find those in ventricles of the brain and in the spinal column or the central canal of the spinal cord of the brain. And again, they are responsible for creating, monitoring, fine-tuning the cerebral spinal fluid. In the peripheral nervous system, satellite cells are also called amphicytes. They surround the cell bodies of the peripheral nerves, and their function is still being decided, but we usually group them into the same category as astrocytes, and that they provide support for the nerves and the neurons. Of course, a macrophage is in the peripheral nervous system. That would be equivalent to a microglia in the central nervous system. So a macrophage would take care of any pathogens. And then we've already talked about Schwann cells and their role in myelin. So that's it for the lecture. Go over this material, answer the questions in a thread on Blackboard, and then discuss any questions that you might have associated with the lecture. You've probably noticed after watching the lecture, maybe looking through the book, that I did talk about some things that weren't necessarily directly covered in the book. But I wanted to give you a bit more of a background in terms of the cell structure to help you understand neurophysiology, because again, it is important to understand anatomy and the structure of the neurons when we're talking about the physiology.